All right. So like I said, this unit is about cell reproduction, human reproduction, and also basic genetics. So we'll start today by talking about cell reproduction. So we know cells have to reproduce for multiple reasons. When is it important that cells are reproducing? Situation many of you are in now at this time of your life. Most of you are growing, right? So to grow, you need new cells. And so those cells come from pre-existing cells. And so growth requires the production of new cells. If you get an injury, if you break a bone, you get a cut on your skin, new cells need to be created to fill in that damage. Your blood cells are constantly dying off, and you need to produce new blood cells to replace them. Okay, your skin is always cells in the surface of your skin are dying and being replaced by new cells. So cells need to be able to reproduce, to replace old cells, to grow new cells. Also, in some organisms, for asexual reproduction. Some organisms reproduce by their cells just sort of splitting and forming a new organism. And typically, our cells reproduce when necessary and then stop reproducing once they've fulfilled their role. Cancer, the disease of cancer, is actually our own body cells, which lose the ability to stop reproducing. And the controls on cell reproduction are broken for some reason. And the cells reproduce, forming tumors. Tumors are masses of cells that are our own body tissues. But for some reason, the cells have lost their ability to control when they're dividing. And those cells can spread, and they can cause disruptions in various body systems. So cancer is really a disease of cell reproduction. So the process that controls cell reproduction is called the cell cycle. The cell cycle is a process cells go through that allows them to divide and reproduce when necessary, and to just go about their usual function when new cells are not needed. And so this is what it looks like. We don't need to worry too much about the details, but the cell cycle includes the cell growing in size, its DNA replicating, and then it's splitting into two. And that process of the cell splitting into two is called mitosis. And that's what we're going to be talking about. Mitosis is cell reproduction that keeps the number of chromosomes the same. A typical human cell has how many chromosomes? Any idea? You're very close, Jason. It's actually double that. We have 23 pairs of chromosomes for 46 altogether. Okay? Some people do have 47. Some people have extra chromosomes. Some people can be born missing a chromosome. And we'll talk about some of those situations as well. So chromosomes, when we talk about chromosomes are small sort of packages of DNA and proteins. Our genetic information is stored in our chromosomes. Each cell, for the most part, in our bodies has basically the same genetic information. A cell in your brain has the same DNA as a cell in your quadricep muscle, which has the same DNA as a cell in your small intestine. Basically, every cell has a complete copy of all of the genetic information. We'll talk about an exception to that later on. And when the cell is sort of going about its normal business, these chromosomes kind of unwind and are just kind of this thread-like material inside of the nucleus of the cell. But when it becomes time for the cell to divide through mitosis, this unwound ball of genetic material winds up into these little packages that we call chromosomes. Often, this is an actual electron microscope image of chromosomes. They often are drawn as these X shapes because they have two halves and they're bound together. So we can see here, this is a chromosome. This is a chromosome. And they're held together with a little piece called the centromere. And if you look at a single double-stranded chromosome such as this, each half of that chromosome is identical. They're exactly the same. So if you have a gene here for brown hair, you have a gene here for the exact same brown hair, and so forth. Where do we get our chromosomes? Yes, we get 23 chromosomes from each parent. Okay, it's always, for the most part, 23 from each parent. 23 from the mother, 23 from the father. And they're in pairs. They're called homologous pairs. They're kind of partners. So in homologous chromosomes, like in, let's say, my third chromosome, one of them came from my mother, one came from my father. They have the same kind of information, but I may have inherited a different gene from my mother than I did from my father. And that's how sexual reproduction works. Mixing of chromosomes from each parent results in variation. You're not identical to either of your parents. You have a mix of traits from each of them. We also know you're not identical genetically to your brothers or sisters, okay? Because you inherited different chromosomes from each parent than your brothers or sisters. What's the only exception to this? Could you ever have identical chromosomes to a sibling? Yeah, identical twins have identical chromosomes. And we'll talk about why that is eventually. So
when you say that you'll never be like exactly the same unless you're an identical twin, does that go for like, because I know some mental things are genetic as well. Does that go for that as well?
Yeah, so anything that is encoded in our DNA, we inherit from our parents, right? Because that's where we get our DNA. And so just like a person may inherit their eye color from a parent, they could also inherit any other genetic factor, like anxiety disorder, for example, or height, or sometimes maybe like musical ability or some musical, So the question becomes then, what factors are genetic and which just sort of develop over our lifetime? And that's a question scientists are always asking. Like, how much of my food preference is just because of the foods I ate as a child and how I was raised versus is that somehow encoded in my genes that I really like broccoli? And so scientists ask those questions. For some things we know and some we don't. So when we talk about chromosome number, the number of chromosomes, we just said the typical number of chromosomes in humans is 46. That's called the diploid number. That's when the chromosomes are still with their partners, with their homologous pairs. But in sexually reproducing organisms, some cells only have half of the chromosomes. What types of cells might these be? Why would a cell have half the number of chromosomes as a typical cell? Yeah, reproductive cells. We call them gametes. In humans, we call them sperm and egg cells. Right? They don't have 46 chromosomes. They have 23 each, because when they come together in fertilization, then you're back up to the typical number, 46. So that is called the haploid number. Gametes, sperm and egg cells, reproductive cells, they actually have half the number. So a human sperm cell typically has 23 chromosomes. A human egg cell typically has 23 chromosomes. Okay. The number of chromosomes is not really a meaningful number in terms of comparing species. Like sometimes you'll say, well, humans have 46. That means, a plant must have 5 because they're simpler, maybe. But that doesn't really mean anything. It's just over evolution what has arisen. For example, a horse has 66 chromosomes. Okay? A cotton plant has 52. Okay? A housefly has 12. Human have 46. A monkey has 28. It just doesn't, it's not a number that really means anything. So what we're going to talk about is the process in which a single cell reproduces to form basically 2 genetically identical cells. And that process is called mitosis. And we're going to go through sort of the steps of mitosis, how it happens. Here's what it looks like. You're going to look at slides either today or Monday that show some cells that are in the process of mitosis. And you're going to have to try to identify what phase they are in. So this is the overview. It's a process that's continuous, but we often split it into what we call phases to just talk about it. And the phases are called prophase, metaphase, anaphase, telophase. There's also interphase. Okay? And what we're going to talk about is what's going on inside the cell in these different stages of mitosis, in these phases. So let's start with a phase that's called interphase. And I'll try and give you hints to remember these. Interphase, if you remember the I, it's kind of an in-between phase. The cell's not actually dividing. So it's not part of mitosis, really. It's after mitosis finished, then the cell's in interphase. It's just hanging out, doing its thing. Being a liver cell, being a brain cell, being an intestine cell. That's interphase, the in-between. And this is when, if you look at a cell in a microscope, you can't actually see the chromosomes. They're all in that unwound ball of string that we call chromatin, like this. So you don't see individual chromosomes like X's or those condensed half X's. It's just like it's all spread out. But when the cell is then going to go through mitosis and it's going to divide, this is when the DNA actually is copied. Obviously, if you're going to make an identical cell, you need a whole copy of all of the DNA to put into the new cell. And so the DNA needs to be what we call replicated. The chromosomes are replicated. So this whole strand ball of genetic material gets copied using enzymes and so forth in the nucleus. And then the cell actually starts to divide. This first step of mitosis is called prophase. Think of it as prepare. The cell's preparing everything so it can make 2 identical copies. So the DNA that was in that unwound ball of string we call chromatin starts to wind up into actual visible chromosomes. So, in diagrams, we kind of draw as a nice little perfect X. It doesn't really look like that. This is what it really looks like. Okay? But we can see that unwound stuff is condensed, and we can actually see individual chromosomes. When this phase is going on, the nuclear membrane that holds the genetic material actually dissolves, disappears. It's gone eventually. And so here you can see we kind of drew it as a dotted line. Here it's gone. You can't see a nuclear membrane in this cell. It disappears for mitosis. It'll reappear later on. The chromatids become visible. And these fibers start to form. They're called spindle fibers. They look like strings that are starting to stretch across the cell. So the chromosomes now are ready. And the cell is able to move on to the next phase called metaphase. In metaphase, the chromosomes line up single file in the middle of the cell. Think about metaphase middle. We could see that here. Those chromosomes are all lined up right down the middle. It's called the equator of the cell. So obviously this is a cell that only has 4 chromosomes. In a human cell, there would be 46 of those chromosomes lined up single file. The chromosomes are attached now to those spindle fibers. Think of them as like ropes that are attaching to the chromosomes. Jordan, you have some notes to write here. And I gotta focus here. So the chromosomes line up in the equator right on the middle. And then the cell moves into anaphase. Anaphase, think away or apart, because these double-stranded chromosomes split apart and they start to get pulled apart from each other, heading to different sides of the cell. So the sister chromatids. Oh, great, thanks. So the sister chromatids split and start getting pulled apart by these spindle fibers. Again, you could see it actually happening in a real cell here. These chromosomes were on the equator, but now they're getting pulled apart. All the way to opposite ends, the poles we call them. Once they've been pulled all the way to opposite ends of the cell, we move into telophase. This is when it's forming 2 new nuclei. So the nuclear membrane appears, one surrounding each set of chromatids that got pulled apart. You can see a new nucleus starting to form here. Here I can see it again. These chromosomes got pulled apart, and now they're on opposite sides. And this is also when, so at this point you have like one cell, but with 2 nucleuses in it. And each of those nucleuses has a full set of chromosomes. The last step is the cell splitting into 2. Each of those new cells is going to have one of these nucleuses. That process of the cell itself splitting, so mitosis produced a copy of the nuclear material, and then cytokinesis actually splits the cell into 2. It happens differently in plants versus animals. In animal cells, because their outer layer is just a flexible cell membrane, it basically kind of pinches in and separates into 2 cells. And we could see that here. Cell membrane pinches in, and now we have 2 cells. In a plant cell, it's a little bit different. Because a plant cell can't just like pinch in because it has that cell wall around it. Instead, in a plant cell, a new cell wall starts to form in the middle, and it kind of grows in and solidifies until it splits this completely into 2 cells. The end result's the same. Final product is we have 2 cells. They each have an exact copy of the genetic material, so the cell has basically copied itself. It's replicated through this process of mitosis. So just to sort of reiterate, here's the process again. So we start at the top. So to remember the phases, you can remember I push mom's angry tiger carefully. You don't want to push an angry tiger without regard. You need to push it carefully. I push mom's angry tiger carefully. Interphase, prophase, metaphase, anaphase, telophase. So here we are. Interphase, a single cell, genetic material all unwound. When it's in that phase, the genetic material copies itself. Then in prophase, it's preparing. The chromatin winds up into visible chromosomes. The nuclear membrane disappears, and the spindle fibers start forming. Those chromosomes move in metaphase to the middle of the cell, single file, like we see down here. They're all in a line. They're attached to spindle fibers. That's metaphase. Those spindle fibers start pulling the halves of the chromosome apart, and that's anaphase. They get split apart from each other and start moving to opposite sides of the cell. Once they reach opposite sides, a new nucleus will form around each set of chromosomes. And then the cell itself splits. And our end result are two cells that have the same number of chromosomes as our original cell. They're identical copies. They're genetic clones of the original cell. We call the original cell the parent cell. We call the two cells that are created daughter cells. Those are the terms that are used. If we look back here, again, in summary, chromosomes condense, go to the middle, get pulled apart from each other, new nucleus forms, two new cells form. There's what it looks like in actual under a microscope. And the end result, identical cells.